Assalamualaikum warahmatullahi wabarakatuh. I am Nur Ina Shahira binti Ahmad Sobri will be presenting my final year project, which is the review entitled Oral and Dental Applications of Curcumin from Curcuma longa turmeric on its antibacterial and antifungal activities. Did you know that, according to the Institute for Health Metrics and Evaluation in the Global Burden of Disease study in 2017, globally, there is an approximate of 3.5 billion people suffer from dental caries, mostly of permanent teeth, which makes dental caries one of the most common and untreated diseases in the world. That brings us to the common causative agents or pathogens that cause oral infections, which are Streptococcus mutans, Staphylococcus aureus, Escherichia coli, and Candida albicans. The problem statement of this review is commercial toothpaste and mouthwash sold in the market mostly contain chemicals such as fluoride, triclosan, zinc citrate, and chlorhexidine as whitening and antibacterial agents. So, the usage of these chemicals is only restricted and limited to certain doses to prevent toxicity and side effects to the consumers and to avoid microbial drug resistance. The use of these chemicals also has some disadvantages, which they can cause taste disturbance, oral ulcers, tooth discoloration, and allergic reactions after usage to some people. Moving on to the significance or importance of this study. This review provides essential information on the antibacterial and antifungal activities of curcumin to encourage the development of turmeric curcumin-based oral products in the future. This is because curcumin is a compound extracted from natural plants, turmeric, so it has the potential to be used as an alternative or substitute to chemical ingredients in commercial oral products. Firstly, let me start with some general information on the plant of interest, Curcuma longa, or turmeric, or known as kunyit in Malaysia. Curcuma longa comes from the native family of Zingiberaceae that can be found in tropical countries like Indonesia, Malaysia, China, Brazil, and India. Traditionally, it is used as spices and medicines to treat hematuria, jaundice, eye infection, and inflammation. The rhizomes of turmeric is mainly consisted of curcumin, which possesses a wide range of biological activities such as antibacterial, antifungal, antioxidant, anti-inflammatory, and anti-cancer activities. However, in this review, I will be focusing on only their antibacterial and antifungal activities. I'm going to explain on two oral or dental diseases, which are first, dental caries caused by the oral bacteria Streptococcus mutans, and second, oral candidiasis caused by oral fungi Candida albicans. To begin with, dental caries or tooth decay is defined as the destruction of dental hard tissue caused by microbial fermentation of carbohydrates. First, Streptococcus mutans and other acidophilic and acidogenic bacteria will colonize the teeth surface in the presence of sugar or carbohydrates that we eat. Then, these bacteria will ferment the carbohydrates to produce organic acids, which will then attract many other acid-loving bacteria to form biofilm. This will lead to the shift in the equilibrium of minerals and oral microbiota, which will eventually cause dental caries spotted by white lesions on the tooth enamel, dentin decay, tooth pulp, and root infection. The next oral disease is oral candidiasis, which is identified by the growth of fungi and penetration of tongue superficial tissue and the mucosal area. After Candida albicans had attached to the oral mucosa to form biofilm, it will change its state from non-pathogenic budding yeast form to hyphal form in order to invade and penetrate the oral tissues and causing oral candidiasis. There are many types of oral candidiasis and each of them varied in terms of symptoms and characteristics. One of them is acute pseudomembranous candidiasis detected by the presence of curdy whitish yellow plaque that can be removed by gentle scraping, which is shown in this figure here, and Candidiasis with the symptoms of ulceration and crusting of the lips. In order to prevent or control these oral infections, the antibacterial mechanism of curcumin on Streptococcus mutans and antifungal mechanism on Candida albicans are reviewed. First, the antibacterial mechanism of curcumin is summarized in this figure. Curcumin acts on or inhibit Streptococcus mutans bacterial cells by several mechanisms, such as by the inhibition of FSG660 protein, ATPase gene, SRTA, GBPB, SCRA, SCRB genes, and FTIF enzymes. They also downregulate the adhesins factors GTFB, C, and D that aid biofilm formation of Streptococcus mutans and limit the expression of quorum sensing, to be specific, the COMCED and LUX-S systems. Next, the concept of antifungal mechanism of curcumin on Candida albicans is basically similar to the antibacterial mechanism, but with the involvement of different types of genes. Firstly, curcumin will increase the reactive oxygen species ROS, which is toxic to Candida albicans fungal cells, thus killing them. Other than that, curcumin downregulates the adhesins genes ALS1 and ALS3, hyphal formation gene TUP1, and upregulates ALS5 and AF1 genes associated with the aggregation of cells. The key point to inhibit the growth of both bacterial and fungal cells is to prevent the progression of biofilm formation, which is the virulence factor of most bacteria and fungi. Now, we move on to the next section, which is the oral and dental application of curcumin from turmeric. In this review, I focused on the three most commonly used oral care products among consumers, which are curcumin mouthwash, toothpaste, and oral gel, and one curcumin product used in dental procedures, which is subgingival irrigant. All these tables here show several studies that have been done in the past years relating to the use of curcumin in mouthwash, toothpaste, oral gel, and subgingival irrigant. To conclude, all findings mentioned the effectivity and safety of curcumin-based oral products in treating different types of oral infections. After reviewing this topic, I found several research gaps, which is first, most studies only focused on the antibacterial and anti-inflammatory activities of curcumin on oral pathogens, but the studies on its antifungal activities are very limited to only Candida albicans. Next, the application of curcumin in oral products other than mouthwash, toothpaste, oral gel, and its usage in dental procedures is barely reviewed and discussed in the journals. Moving on to the conclusion. To sum up, curcumin possesses strong antibacterial and antifungal activities to inhibit bacterial and fungal pathogens like Streptococcus mutans and Candida albicans. Thus, it has high potential to be used as a substitute for harmful chemicals in commercial oral care products like mouthwash, toothpaste, oral gel, and subgingival irrigant. My recommendation is that more research is required on the antifungal activities of curcumin on oral fungi and also its application in dentistry in the future. So these are my list of references. To end, I would like to thank my supervisor, Professor Madhya Dr. Zaida binti Zainal Arifin, and the Faculty of Applied Sciences, UiTM Shah Alam Selangor. Thank you for listening and have a nice day.